All right, one virus we're going to spend a little more time on here is going to be the human immunodeficiency virus, or HIV. Because as future healthcare providers, this is a virus that you could come across during your time as a healthcare provider. And in terms of that HIV, transmission classically associated with HIV is going to be sexual transmission, although it is also transmitted via blood products, all right? And it's also transmitted mom to baby, okay? So in terms of viruses and transmission, some viruses have one mode of transmission. Other viruses can have multiple modes of transmission. HIV is a good example of a virus that can be transmitted via multiple different pathways, okay? And in terms of the HIV virus, all right, one thing about the HIV virus that makes it a little bit unique is it's a retrovirus, all right, which means it has an RNA genome and it needs an enzyme in order to replicate in a host cell, all right? That enzyme is reverse transcriptase, okay? So in terms of our HIV virus, all right, we've got our RNA as our genetic material. The triangle here, that's our capsid that contains the HIV virus. Here we'll make them orange. This is that reverse transcriptase because synthesizing DNA from RNA is not something that our cells do. So that virus has to bring in that protein that it needs, okay? So that reverse transcriptase is a protein. It's brought in as part of the virus particle itself, okay? The HIV virus is an enveloped virus. All right, so in terms of that, envelope viruses are a bit more susceptible to various cleaning reagents. So they're easier to clean, say, than some other viruses that are naked viruses. The envelope there is going to come from the host cell. And then in terms of that, we're again going to have spikes that are going to come off of the protein. And these are going to be, or off of the protein, off of the virus. And these are going to be what are recognized in the host, in this case our eukaryotic cell, all right? And specifically, that spike on the outside of the HIV virus is going to be recognized by immune cells. So what the HIV virus does, in effect, is it destroys the host's defense system, all right? And what that means for folks who have the HIV virus is that as that virus destroys more and more of their T cells, it results in their not being able to fend off other diseases that typically your immune system can handle, like something as easy as the flu can result in death with someone who has an HIV infection, all right? Yeast is another organism that can kind of take over, all right? Various bacteria can also result in the death of the patient, okay? And in terms of that, they're also more susceptible to a certain type of cancer known as Kaposi's sarcoma, all right? That's because that HIV virus is destroying part of the immune system that is involved in fending off that particular type of cancer, all right? So really the virus doesn't kill the person. What ends up killing the person is another disease, okay? And that is really what the distinction is between HIV, which is the virus, and AIDS, okay? AIDS is the syndrome that results from HIV infection, okay? And really what ends up happening is the immune system is weakened, and that allows a lot more opportunistic infections to occur. All right? So in terms of that HIV virus, all right, it's an RNA genome, it's an envelope virus. There's a difference between the virus itself and the syndrome that results in the death of the patient, all right? There are a number of stages that result from HIV infection. The first stage, the person, when they're exposed to the virus, may end up having symptoms of like a cold. They've got maybe a slightly elevated temperature, they kind of don't feel well. But in terms of that, it tends to be relatively short. It resolves like the flu or the cold would, and the person may not realize that they've been infected, all right? One of the key things that the HIV virus can do that not all viruses can do is that it can integrate into the host's genome. All right? And that ability of that virus to integrate into the host DNA cell is what gives rise to the big spectrum we see of somebody initially being infected with the virus versus actually coming down and showing the symptomatic, the symptoms associated with AIDS, okay? That latency period essentially depends on where that HIV virus integrates into the host genome. If it integrates in a place where the genes aren't transcribed or the genes aren't used, you're not going to get a lot of replication of that HIV virus, and it's going to kind of lay dormant, all right? It's not completely dormant because even though it's integrated into the DNA, folks who have the HIV virus still can pass it on to other sexual partners, all right? But typically their viral load is a lot lower, okay? When the viral load is higher, you're much more likely to transmit it, okay? This is why some people can go 10 to 20 years before they show any symptoms of AIDS, all right? Because it depends on where that virus integrates into the genome. All right, so in terms of that, HIV is a little bit unique because it has that ability to integrate, all right? And in terms of HIV, I've got another video after this that I'm going to talk a little bit more about treatments with HIV. And in terms of treatment with HIV, where you're looking at treating the virus, okay, is going to be in places like interfering with the ability of that virus to integrate, or here, interfering with that virus's ability to make a DNA copy from an RNA copy, okay? So in terms of that, we'll talk a little bit about the different inhibitors associated with the HIV virus and potential treatments as well, all right? So if you have any questions, please